Welcome to Grow Bio channel. In today's video, we're gonna see about gene regulation in prokaryotes. Overview. Gene regulation. The cell prevents wastage of energy by switching on and switching off the gene, that is coding part of DNA. History. In 1951, Barbara McClintock, she showed interaction between activator and dissociator genetic loci in maize. And in 1961, Francois Jacob and Jacques Monod studied gene regulation of lactose catabolism in Escherichia coli. Operon. What is operon? Operon is the genetic unit which has adjacent gene, structural gene, non-coding regulatory gene, etc. Structural gene, for example, LAC-Y, LAC-Z, LAC-A gene, etc. And non-coding regulatory gene, which doesn't code any protein, for example, promoter, operator gene, etc. The cluster of genes work under same promoter is said to be operon. Stages of gene regulation. There are many stages to regulate the gene. Gene can be regulated from transcription process to the post-translation modification process, such as chromatin domains. There are some molecules that change the chromatin domains itself so that it can't be transcribed or translated. Next is transcription. There will be altering in transcription factor molecules to regulate the gene regulation. Either it can promote the transcription or it can prevent the transcription from happening. Next is translation. The translation process. Here, gene can be regulated by changing some molecules such as sigma factor, which is responsible for knowing promoter sequence in mRNA. Such sigma factor can be altered so that translation will not occur and there can be change in ribosomes, etc. and post-translation modification. When the protein changes its structure, then its function will change. So, when post-translation modification is regulated, it means the gene is regulated. Next is mRNA degradation. When mRNA is not produced or mRNA is degraded, then translation will not occur. So these are certain stages of gene regulation. Next, gene regulation in prokaryotes. In response to the environmental change, the presence or absence of certain compounds, in prokaryotes, the gene is switched on or switched off. There are two levels of gene control. First is transcription mechanism control, that is control of mRNA synthesis. Next is translation mechanism control, that is control of protein synthesis. The gene expression is of three types. Constitutive gene expression. First we can see about constitutive gene expression. Here the gene is expressed constitutively, that is continuously. Continuously this gene will be active. Okay, without any signal, constitutively this gene will be active and producing protein. This will be responsible for certain proteins which is needed by the body all the time. Next is inducible gene expression, that is induced. Okay, the gene is expressed only on presence of substrate. Here the substrate will act as the inducer to switch on the gene. For example, it will be only available in catabolic pathways. For example, lactose catabolism. Here the lactose will be inducer. It acts as the inducer for the catabolic enzyme synthesis. Next is repressible genetic expression. The gene is expressed always, repressed only on presence of byproduct, that is repressor. Here the byproduct itself acts as the suppressor. For example, in tryptophan metabolism, the tryptophan is produced. When required amount is achieved, the tryptophan itself acts as the repressor for its synthesis. Okay, so the repressible gene expression will be available in anabolic pathways. Now we can see about response of gene expression. There are three types of response. First is dependent response, a type A response. Initially there is no inducer. The signal will not be there. When the signal persists, then the reaction will occur. When the signal is down, then the reaction will not occur. Again, when the signal comes, the reaction occurs. This type is known as dependent response, that is signal dependent response. As you can see in that picture, with respect to time and rate of genetic expression, when the signal persists, the reaction goes up. And when signal is not there, it is down. Again, when the signal comes, it is up. It is known as dependent response. Next is transient response. It is type B response. Even though signal persists, the reaction comes down. That is, transient increase of expression occurs in the organism. When the signal comes, the transient increase of genetic expression will be there, and even though the signal persists, it comes down. Again, when the signal comes, there will be transient increase in the genetic expression. Even though the signal persists, again it will come down. So, at the starting of the signal, it will be high. Even though the signal persists, it will come down. It is known as transient response. Next is indefinite response, the type C response. Once the signal comes, then there will be continuous gene expression. Even though the signal stops, the reaction will not end. So this is known as indefinite response. Next is control mechanism of gene expression. There are two types of gene control mechanism. First is positive gene control and next is negative gene control. Now we can see one by one. Positive gene control. The product of the regulator gene acts as the inducer, is required to turn on the reaction to express the structural gene. For example, in lac operon, there is a cap site. Okay, that cap site is responsible for cap pathway, that is catabolic activator protein, okay, which will be combined to CAMP, cyclin adenoside monophosphate. When this complex interact with the cap site, then the lac operon will be induced, so the reaction will occur very fast. So this is called as positive regulation. Whereas in negative regulation, the product of the regulator gene acts as the repressor, is required to turn off the structural gene reaction itself. For example, in trip operon and lac operon, it is a repressible protein. Okay. In lac operon, the I gene will produce the repressor protein continuously, whereas in TRP operon, that is trip operon, the tryptophan will act as the repressor. Okay, the byproduct of tryptophan metabolism will act as the repressor for the reaction itself. So this is known as negative regulation. In next video, we can see about trip operon and lac operon. Thank you. Let me know if you have any doubts by commenting in the comment section below. Subscribe and please press the bell button below. Thank you.